Assistant Professor, Faculty of Pharmacy, to represent the sapling to Dr. Mohammad Shahar Yar Khan. Mohammad Shahar Yar Khan, Professor, School of Pharmaceutical Education and Research, Jamia Hamdard, New Delhi. A huge round of applause, everybody.
Now I'd like to invite Mr. Prasanjit Paul, Associate Professor, Faculty of Pharmacy, HOD Department of Pharmaceutics, to introduce Dr. Mohammad Shahar Yar Khan. Please, sir.
Jain, good afternoon to all of you. It gives me an immense pleasure to introduce in front of you Professor Dr. Shahar Yar Khan from Jamia Ahmedabad, who is our first speaker in the second scientific session. Professor Shahar Yar Khan has done his graduation and post-graduation from Jamia University, New Delhi in 2005 and 1995 and 1997 respectively, and done his doctoral program from the same university in year 2002. He is well familiar with the in silico design of drugs apart from the traditional mode of drug design. He has experience in troubleshooting chemical reactions like side products and analyzing impurities. He has been associated with collaborative research with TAACF, Southern Research Institute, Alabama, USA, Riga Institute for Medical Research, Leuven, Belgium, and National Cancer Institute, USA. To his credit, he has successfully guided 25 PhD scholars and 27 master students. Dr. Mohammad Shahar Yar is involved in synthesizing various heterocyclic moieties such as benzimidazole, benzthiazole, pyrazoline, pyrimidine, etc., and their biological evaluation, which includes antidiabetic and anticonvulsant, diuretic, and anti-cancer activity. He has currently been working on the design and development of benzimidazole derivative as anti-cancer agent, and has published as many as 160 research articles in journals of national and international repute, and has 37 monographs to his credit. His current H index is 45 with about 7,100 citations. He has handled several research projects of more than 1.5 crore rupees, funded from various funding of authorities like ICMR, UGC, AICT, CCRUM, etc. Recently, he has been awarded AICT Viseswara Best Teacher Award 2020. Dr. Shahar Yar has immensely contributed to the growth of Jamia Hamdard, in particular in its pharmacy school. He has played an important role in the securing #1 NIRF ranking to the institute in 2017, 18, 20, 21, and 2022. In addition to his achievements and academic and research activities, he is serving as Deputy Director of Jamia Hamdard IQAC Cell, Deputy Proctor of Jamia Hamdard, and member of Central Admission Committee. He has also served as Warden of International Boys Hostel in charge, University Guest House, etc. As an additional responsibility, he started the BTech Food Technology program in Jamia Hamdard University in the year 2016. He was coordinator of IGNU program of food technology during the year 2016-17 and 2018. Now I welcome Dr. Khan to give his delivery. Thank you, sir. थैंक यू। पॉइंटर दोगे मुझे कोई? नहीं। So very good evening, not afternoon because it is already 3:45, and definitely whenever we are going to have any kind of session just after lunch, it is very difficult to handle the people because any conference starts with the inaugural and finishes with the lunch. So I could see the good number of the students who are present here. Anyways, I appreciate that you people have still waiting for the evening tea. That's why you are here. So I am this kind of person, so don't take anything seriously. So the topic for the today's talk is drug repurposing. I don't know how many of you are passed out, are completing the B Pharm. And the topic is basically the newer one as compared to the situation, because this terminology has been arose in 1920, but recently during the COVID time, it has got the momentum. Reason behind that discovery and development takes longer time for a particular medicine, but during the COVID time, we need a medicine within a very short period of time. Because of that, this repurposing, the drug repurposing concept was further reignited so that the minimum efforts can produce the maximum efficacy. In the normal case, I'm just making this is Bhumika, but I will go a little faster for that, just to make you understand that what is the drug repurposing. I don't know whether you people have studied it or as of now or not, because you are in B Pharm, and up till B Pharm final year, you people may not be acquainted about these concepts. So basically, the drug development is a long process, which takes at least 15 to 20 years. By the time, if any emergency situation comes like COVID pandemic, within that period of time, we need a very recent drug. And as per the process, whenever a drug is manufactured, toxicity studies, formulation studies, and it requires many aspects. And after that, we realize that we failed. Many of the time, the drugs which have been marketed has to be withdrawn from the market because of the side effects, lethal side effects. So in that situation, so the concept of the drug repurposing was to use of a particular medicine which is still being used for the treatment of X disease can be used for the treatment of Y disease, which has not been utilized further. Because we had assumed that this drug, and established, not assumed, initially we assume and then we establish that this drug is basically useful for the treatment of this particular disease. So by the time we came to know that this drug does not have the toxicity or has the very little toxicity. This has been established. So this timeline has been saved. This timeline, which we are supposed to be development, then conception, then development, then formulation study, pharmacological study, toxicology study, all these steps has already been established because this drug is already being used for the treatment of a particular disease. So this time has been saved. The amount which is supposed to be invested, that is also being saved in this period. So now what is to be done? We have to only procure the data of the existing medicine, and we have to evaluate by a suitable means that whether this drug is applicable or useful for the treatment of this disease or not. How can we achieve this? Simply by using the more modern techniques or advanced techniques. In silico screening is there, computer-aided drug design is there. Now we know the protein structure, and we know the structure, which is already established. So by virtual screening, we can overlap the thousands of the molecule onto the protein ligand, and whichever is completely fits, that may be used for this kind of the treatment, which was being used for the X disease. Now the proteins of the various other type of the diseases have been procured from the PDB, and the molecule which are we are assuming that it could produce that kind of activity are superimposed, and this is the simple concept of the lock and key. The receptors which are present in our body are locked.
God-gifted locks. And the medicines, the molecules are basically the key. So when it is appropriately fit, it can open and close the lock. So it does not mean the key of a particular lock can open only one lock. Sometimes it opens, there is a master key also, which can open the maximum number of the locks. And there are fair possibilities. The normal key can also open out of 1,000, 5 or 6 locks also. The same concept is applied for the repurposing, because the drug which is being used for the treatment of this disease, why not it can be tested for the evaluation of the other disease. So this is the basically theme. Now I shall go by the PPT because the number of the PPTs are more, and I will skip, but I wanted to just explain what is the basic theme of this talk. Please, ma'am. पीछे जाएंगे, कंट्रोल मुझे दे सकते हैं क्या? इसे पीछे, हाँ, इसके पीछे भी एक स्लाइड होगी, इसके पीछे एक स्लाइड, इससे ये तीसरी स्लाइड पे हैं आप. चलिए कोई बात नहीं, मैं यहीं से शुरू कर दूंगा, कोई हर्ज नहीं है. Yes, this is the first slide. Let me start from here only. So finding new molecule entities by the traditional or the de novo approach of the drug discovery is lengthy and time consuming and expensive. Whatever I have just said, that it has been there, so I will be a little faster in that case. Drug repurposing is also known as drug repositioning also. It has several names, but commonly it is known as the drug repurposing, as well as it is also known as commonly as the repositioning. It is a process of identifying new therapeutic uses of the old or existing medicine, which I have just explained. It is an effective strategy to discovering or developing the drug molecule with new pharmacological and therapeutic interaction. In recent years, many pharmaceutical companies are developing new drugs with the discovery of the novel biological targets by applying the drug repositioning strategies in drug discovery and development program. So major advantage, tell you next please. Finding new molecular, and this study is highly efficient, time-saving, low cost, and minimum risk of the failure. Why? Because most of the time we find that the drug which have been developed and later it is having the toxicity studies. Rofecoxib, I don't know how many of you are aware, the Ranbaxy Pharmaceuticals has developed Rofecoxib. And during 1997-98, it was banking on it, that was a selective COX-2 inhibitor. But unfortunately, the drug during the clinical trial for phase 4, post-marketing surveillance, failed because it was causing the cardiac arrest. And ultimately, they were supposed to withdraw this molecule. And when this drug was withdrawn, that was the basic reason of the collapse of the Ranbaxy. It was sold off to some other, and right now it is Sun Pharma. So basically, investment is huge. The returns are not guaranteed. So it is very risky business because it requires 15 to 20 years and infinite amounts of the money which has to be invested. And outcome, even after the marketing, there are fair chance that post-marketing surveillance, the side effects may be so grave that the drug has to be withdrawn, and that had happened. So if any drug which is already being established for the 20 years, which is being used for the treatment of the X disease, that means all the lethal and the other parameters have already been established. So that means it can be used for the treatment of the other disease as well. So please next slide. मैं थोड़ा सा ड्रग के नाम बताने पे ज्यादा ध्यान दूंगा. This important. According to the recent estimate, pharmaceutical industry have significantly placed the market for the repurposed drug is $31.3 million by 2020. And it is expected this growth will go up to the $50 billion by 2025. The first example of the repurposing was noted in the 1920, but it does not play a very significant role. And hardly 10 years back, or we can say 7 years back, this methodology is picking up very faster because the development of the new medicine is a very costly affair. So recent years, the drug repositioning strategy has gained considerable momentum, with about 1/3 of the new drug approvals correspond to the repurposed drug. Means we have to go for the agency for the approval. Although all the other parameters are established, no need for the toxicity data, but we have to prove that this drug is effective against this treatment of this kind of disease. So first we have to carry out the CADD and computer-aided drug design, as well as the in silico screening. And after that, we have to give to the patient just to check the efficacy. You don't need to check the side effects because that has already been established. Next slide, ma'am. नेक्स्ट स्लाइड मैम. ये तो आगे जाना है ना, पीछे जाएंगे एक स्लाइड और 6 पे जाएंगे आप. मैं इसका वो दे दूं क्या आपको? पीडीएफ है, ये पीडीएफ चलने में मुश्किल हो रही है ना, मैं इसका वो वर्जन दे दूं, पावर पॉइंट दे दूं. मैं समझ रहा हूं. बस यही है, पावर पॉइंट वाली ले लीजिए, इसमें कुछ करके आप ले लीजिए. मुझे पहले वापस advancement of the bioinformatics. Now we have to learn the new techniques. Whatever we are studying in the pharmacy, that is good. But if we do not know about the bioinformatics, because various tools are available, so in which has a large data bank of the various medicines, so that if we can pull them up and we can PDB data bank we can access, because many of these data are already available free of cost. And many of the software which are available, bioinformatics software as well as the drug design software, many of them, Autodoc, VLife, even the Schrodinger, all these softwares are free of cost available for the initial purpose. But once you have established it for 1 month license, and after that you can purchase also if any institute want to. So, but we can go for the free software as well, because the plenty of the softwares are the Swiss ADME is also there, one of them that is working good. So the bioinformatics and the chemoinformatics tools, which provide the biological and structural database, drug repositioning has significantly decreased the time of the cost of the drug development and reduction of the risk of failure. In recent years, the use of the in silico technique, in silico means the way first I am talking about how to find the PDB, the protein data bank, because whatever receptors we have, that has protein. And there is a data bank of all these proteins receptors, which are available, that is actually locks present in our body. And the molecule, various small protein-like molecules are discharged in our body, they are maintaining. But when there is imbalance, so the external medicines has to be given, that is basically act as a key out to them. So they can open and lock. So accordingly, so we can use the artificial intelligence also. Artificial intelligence, as we are knowing today, that we have to go to this place. So we know that within 40 minutes time we can reach, because it is already reading it by virtual, how much traffic is on the road. So that is artificial intelligence. So the similar way, we can predict activity of a drug based on the data available. So for that, we have to feed the data. What the Google is doing, that is feeding the data to the system. So huge data is fed so that we can access that this point is having the traffic and this point doesn't have the traffic. The same concept is to be applied for the drug discovery. So based on the previous data, all the data has to be fed to the system, and we can predict that this drug may pass on through this passage, and we can clear this passage, or we can do that. So the same concept, nowadays the traditional things is not working. So AI is that based on that past experience, we have to feed to the system, and we can predict the data, what could be the outcome of this medicine. So AI is basically, similarly for the sildenafil, which was basically developed for the heart disease, coronary heart disease, and later it was found as the erectile dysfunction. It is being used for the treatment of the erectile. That is called as serendipity, which was not predicted, but it has shown. So this is also a kind of the repurposing. Although what for it has not been made, but it is being used for the treatment of that disease, which was not expected. It was basically made for the treatment of the heart disease, but it is being used for the treatment of the erectile dysfunction. Similarly, the metformin is also a drug which is used as for the treatment of the diabetes, and which is also being used for the treatment of the cancer also. That is also a role of the drug repurposing. Made for some other thing, but being used for some other thing, and nowadays it is in the phase 2 clinical trial. If gets successful, so within a no period of time, this can also be used for the treatment of the cancer. Next slide, please. Normally, whatever I was talking about, in normal approach, that drug development takes 10 to 16 years. 10 to 16 years when we are going from the scratch, and it requires approximately 20 to 25 billion dollars, but the repurposing reduces it to approximately only 1.6 billion dollars, and the time is also from 6 to 9 years for the development, because all the previous activities, toxicology and the establishment of the formulation is being removed, because that has already been established. Next. Next. There are two strategies for the development. One is the on target and second is the off target. Next slide. In on target drug repurposing, we know the pharmacological action. We know the pharmacological action of a particular molecule, but we have to develop what other kind of the activity it can exhibit. For example, minoxidil. Minoxidil, I don't know in which class you people are studying, but minoxidil you must have studied. That is a vasodilator for the treatment of the heart disease. But it has a side effect. What is the side effect? Enhanced hair growth. So that is also being nowadays used for as a repurposing for the use of the hair growth. Keeping in mind that it does not affect the heart rate, if anybody. And nowadays, it is being used as a local application, not by the oral or the injectable. Because it is not going to that much of the systemic circulation as the oral can be. It is only applied onto the portion where we wish to grow the hairs. So that is the advantage of the repurposing that we can establish. It is again a side effect, being used as a medicament, and it can be re-justified by the supporting data, by the clinical trials and other things, which will reduce the time for the development of the medicine, but only those parts will be established that it does not have any other side effect related to the hair growth. Similarly, the other kind, next. Off target is also there where we do not know the pharmacological action. For example, aspirin. We know the aspirin is being used as a non-steroidal anti-inflammatory drug, NSAID. But two other activities have also been found. Nowadays, it is under a clinical trial for the treatment of the colorectal cancer. I believe in my studies for the last 25 years I'm teaching, I don't find that the simplest drug available, any medicine which is the simplest formulation, that is the acetyl salicylic acid aspirin. And the second drug is the paracetamol. These two drugs are simple acetylated product and being used for the n number of treatment of the n number of disease, amongst them the acetyl salicylic acid. Salicylic acid is antibacterial, freely available. COH and OH group are present at the ortho position to the benzene ring, and when acetylation is carried out, OH is converted to OCOCH3, it changes its property and it becomes a medicine aspirin. कहाँ use नहीं हो रही? सबसे पहला use दर्द के लिए. उससे ज्यादा use, it is being used for the treatment of the heart disease, heart patient. Every prescription, you take the prescription, you will find ecoaspirin, 75 mg. That is what? Aspirin. Why it is being prescribed? Not for the pain. Anyone can answer this question? Why it is being prescribed for the heart patient? Because its side effect is, it is a blood thinner. It does not allow the platelet to coagulate. And once the platelets are not coagulating, that means the blood coagulation will not take place and the flow of the blood will be maintained within the arteries. So it is a blood anticoagulant की तरह काम कर रही है क्योंकि platelet को aggregate नहीं. This is a side effect and it is being used as a purpose for the treatment of the blood thinner. Because in case of the patient who are having the heart disease, there is a rupture of that and the coagulation may take place and that coagulation, clot may be inhibit the flow of the blood. So that's why it may go to the brain, it may go to the heart, any place. So this is also being used as a blood thinner. Third use is what? For the treatment of the colorectal cancer. So this is also one of the use. So agar itni simple medicine hai, so that's why we should try to encourage our student to develop a simple molecule rather than a complex molecule. We believe that the structure is huge, having the many binding site may be effective. No. The simplicity is the best thing, I believe, for the human being as well as for the molecule which are being for the treatment of the disease. Simple example, I said aspirin and paracetamol. Paracetamol is acetaminophen. Para-aminophenol acetylation is carried out and it gets converted into acetaminophen, commonly known as paracetamol. Aspirin, acetyl salicylic acid नहीं जानते, एक चीज जानते हैं, डिस्प्रिन. ब्रांड नेम को हम जानते हैं, जबकि एक्चुअल नाम उसका क्या है? acetyl salicylic acid aspirin. Similarly, for the paracetamol, that is acetaminophen. From the name, it is clear. Acetaminophen. Acid means acetyl group. Amino means there is amino group. Where? In the phenol. C6H5OH and para position is NH2 and NH2 is H is replaced by the COCH. इससे ज़्यादा आसान क्या बना सकते हैं? कोई भी कहीं भी बना सकता है. आप अपने घर पे बना सकते हैं. इससे ज़्यादा आसान क्या है? भाई अगर आपको आगे चल के एंटरप्रेन्योर बनना है, आप यही सिर्फ बनाने एपीआई बनाने के इंडस्ट्री डाल लें, क्या बुराई है? कितनी देर लगती है? आपको एक हीटिंग मेंटल चाहिए अगर छोटे स्केल पे बनाना है, एक आरबी चाहिए, एक रिफ्लक्स कंडेंसर चाहिए और 2 आइटम चाहिए जो फ्रीली अवेलेबल है. तो फार्मास्यूटिकल इंडस्ट्री में हम लोग कॉम्प्लेक्सिटीज में उलझे हुए हैं. वी आर नॉट ट्राइंग टू फाइंड आउट अ सिंपल मॉलिक्यूल विच मे हैव अ वेरी गुड पोटेंशियल. दिस इज अ डिफरेंट इशू. नेक्स्ट प्लीज. चलिए. So that was on target and off target. Various approaches are there. Experimental based approach, in silico. Experimental based, I just told you that we have to carry out the various experiment, which is a traditional one, and in silico is the advanced one, in which we have to find the software, bioinformatics software or the other drug design software, which are many of them are freely available, and we can carry out, and based on that we can protein, that is the PDB site, and we can carry out the several approaches: experimental reposition, target screening approach, cell assay approach, animal model approach, and the clinical approach. Next. In contrast, in silico repositioning, or you can call it the repurposing, carries out virtual screening. What is the virtual screening? The thing is not there. We are assuming that we are working onto the body. Virtual means there is a replica available in the system. the look-alike whatever is present in our body is already the same parameter is available into the system. It is assumed. आज के वक्त में credit card क्या है? पैसा कुछ नहीं है, but we are paying. What is that? Virtual money. But basically the same way is a virtual thing. There is the thing, पैसा है, वो आपको देना है, लेकिन उस वक्त आप नहीं दे रहे हैं. The same way, all the parameters which are present in our body has already been fed into a system. And the drug structure which we have drawn, we have not made the drug, but all the expected parameters we have fed it to the system and just inserted the structure, and the structure will automated process will find a suitable binding. How effectively it can bind to the protein. And based on that, we can decide that this structure, the substitution present at this position could be more effective molecule. So this is the concept which is applicable into the in silico. Next. चलिए next कर लीजिए. चलिए, तो this is the one thing, the old concept and new concept. So there is a conflict. So the best thing is the mixed concept. So that's why the mixed approach is a result of the computational method in the validation of the preclinical biological experiments, in vitro and in vivo tests, and the clinical studies. Nowadays, when we are going for the screening of a new molecule, the pharmacological agencies and the animal protective group, they are not allowing to use the animal. So that's why we have to go to the in vitro screening. The cell lines are there, or other methodologies are there. So now we can reduce the use of the animal also, which is highly unethical. Although we are killing the animal unnecessarily, so we are reducing by using this methodology also, and we are using only the established, and by using the in silico methods and in vitro method in which the cell lines are there, or other methodology in which blood and the animals are not required. So both interface can use. Partially we are using the animal where the 100 animals were required, we can reduce to the 10, 20, and based on that, we can give the medicine to them, and their blood can be taken, and we can carry out the screening. So the drug utilization of the animal is significantly reduced. So next, please. So it can be divided. Methodology is drug-oriented, target-oriented, and disease-oriented. This is our choice. Whether we want X type of drug, so we can go for the drug. We can make the substitution into the drug, keeping the basic skeleton same. We are changing the CH3 group, OH group, NH2 group into the molecule. Whereas in the target-oriented, we want to treat brain tumor, we want to treat the colorectal cancer tumor, we want to treat convulsion, we want to treat hypertension. So accordingly, we can decide, because we know the protein structure, and accordingly, we can design the molecule, and molecules are already available. We have to simply give to the data to the system, and the result will come out. They will tell you that this molecule is most suitable for this type of treatment as well. I don't know stereochemistry, you people are aware or not. Stereochemistry play a very significant role. Sometimes C6H5O12, very simple molecule. What is it? C6H12O6. What is it? सब ग्लूकोज कहते हैं, सब ग्लूकोज कहते हैं, फ्रुक्टोज क्यों नहीं हो सकता? लेकिन एक कॉमन है, हमारे दिमाग में बस बैठा यही है. दोनों का फार्मूला एक है, स्ट्रक्चर अलग है दोनों का. एक वन एल्डिहाइडिक है, दूसरा कीटोनिक है, लेकिन हम पहचानते किस नाम से? सिर्फ ग्लूकोज. कभी भी फ्रुक्टोज नाम जब दोबारा तिबारा का होगा तो आदमी को क्लिक होता है कि दूसरा भी बोलना है, वरना हमारे दिमाग में एक आइडेंटिटी बनी हुई है कि C6H12O6 is only glucose. Both having the different role. Similarly, dextromethorphan, levomethorphan, same structure. The only change is rotation of the plane of the polarized light. Dextromethorphan rotate the plane of the polarized light onto the clockwise direction. Levomethorphan rotate the plane of the polarized light anti-clockwise. One is used for the anti-tussive, or other is used as NSAID. Altogether classifications are different. So this is also a role of the stereochemistry, also play a very significant role. So chemistry is very interesting. Why I am emphasizing chemistry? Because I am from the chemistry background. So that's why. It does not mean I am influencing you to have the chemistry, but I must say that the future, I'm not pointing out, or I'm not demeaning any other department, but at the same time, I must say the future is chemistry, believe me or not. Because the pharmacology people have already exhausted all those things, whatever were present. Pharmaceutics people can prepare the formulation if the chemist is providing a new molecule. So that's why the role of the chemist becomes very significant. If we do not have new molecule, the only thing is we can do the repurposing. तभी रिपर्पसिंग है कि हम कोई नई नहीं बना पा रहे हैं, मजबूरी है, क्योंकि पैसा इतना खर्च नहीं कर सकते, कॉम्पिटेंसी इतनी है नहीं. दोनों बातें हैं. तो इसलिए क्या है, अब एक रिपर्पसिंग भी है और एक इसी के बीच घूम सकते हैं, क्योंकि इंजेक्टेबल बनाने की कैपेसिटी नहीं है. ये बात भी, क्योंकि दैट रिक्वायर्स हाई ग्रेड ऑफ द एफिशिएंसी, और प्रॉब्लम हो जाएगी. Syrup, suspension, tablet, patches. These things are easy to handle as compared to the injectables. That is not so easy to handle. So that's why in the institution level, we can do all these kind of things. So the credit goes to the teachers because they are engaged in the research teaching, which is a primary duty. At the same time, they are carrying out the research. So our role becomes more important because we are handling two things simultaneously, and we are preparing the future scientists as well. So we have to orient our student in such a manner they can decide from the beginning that which field they have to choose. I'm not saying that you should choose chemistry, because the difficulty level in the chemistry is to stand into the laboratory is very difficult. Play with the chemicals is very risky. But the person who has the ability to handle all these things, they should come forward with the knowledge of the basic concept of the mechanism. Because most of the time, we know we are forced to learn the structure. I'm not in favor of that. If you should be forced to learn the structure, rather you should be, I request to our fraternity, rather asking the question to write the structure, you should change it to write the highlight of that structural feature rather than the structure. So that at least they can able to know that there is a indole moiety, there is a imidazole moiety, rather than asking the structure. क्यों? सच्चाई ये है कि जब टीचर इवैल्यूएट करने बैठता है तो 90% उसे भी स्ट्रक्चर नहीं पता होते. दिस इज द हार्ड ट्रूथ. मैं एक्सेप्ट करता हूं, कोई करे या ना करे, मैं तो करता हूं कि जब हम पेपर बना लेते हैं, जिस पेपर से आपको पास और फेल करते हैं उसी पेपर को मुझे दिया जाए, मैं फेल हो जाऊंगा. दिस इज फैक्ट. तो वी शुड बी लिबरल. इट डज नॉट मीन वी शुड बी दैट मच लिबरल कि आपने कुछ नहीं लिया, हमें पास कर देना चाहिए. आपको कॉन्सेप्ट हमें बदलना होगा. वी शुड नॉट आस्क द स्ट्रक्चर, रादर देन वी कैन encourage to understand the structure, that what changes are being taking place and why this is showing this kind of efficacy. I never mark based on the incorrect CH3 group or OH group. I simply see the skeleton is correct or not. That is enough to me. आपने नाक को नाक की जगह पर नहीं, जरूरत नहीं है. नाक है या नहीं है वो मेरे लिए इंपॉर्टेंट है. वो अपनी जगह पर या नहीं है वो तो वक्त के साथ सेट हो जाएगी. So that's why this is the concept. I request my teachers also, they have to change, because the writing of the correct structure is not the what you are testing. You are simply testing how good you can mug the structure, not the understand. So that is a different issue, coming back to again because it is not online. चले आओ नेक्स्ट आ जाओ. चलो आगे चलते आओ, जल्दी जल्दी जाओ. टाइम बहुत खराब कर लिया, आधे पे आ जाओ. जहाँ पे टेबल सारी है वहाँ पे बता दिया मैंने सब. ये भी चलो आगे चलो, आगे चलो. These are the various methodology. No need to go. I wanted to go to the tables where I wanted to show what the drugs which are available and being used for the some other things. रुक जाइएगा यहाँ पर, पीछे जाएंगे, एक पीछे ले लें बस प्लीज. 28 पे चले जाएं. Thank you. So what I was talking about, the change took place majorly during the COVID time. So the antiretroviral drug or retroviral, both are the same thing. That is a specific kind of the virus. Effect for the older anti-malaria drug. Chloroquine, everybody must have heard that chloroquine has been used. But there was no specific data available. But there was no other way out. हमारे पास option क्या था? What to do? So that's why we have to try all type of experiment. We are basically experimental animal. But during the COVID period, whatever the vaccine we took, basically that has to be done by the DCGI office, and that has to be established all those the side effects. But the time was so limited. मरना है या यहाँ उम्मीद है बचने की, तो आप किसको चूज करोगे? बचने की उम्मीद पर, जबकि गारंटी नहीं है उसकी, लेकिन एक उम्मीद तो है. मान न लेने पर गारंटी थी कि मर सकते हो, तो इसलिए क्या करना है, हमें लेना ही पड़ेगा. बहुत लोगों ने सर्टिफिकेट बना के काम चला लिया जो अभी बन रहे हैं बेवजह. लेकिन मेरा एडवाइस है, वेयर एवर द सेफ्टी इज देयर यू शुड टेक इट. देयर इज नो हार्म बिकॉज ऑल पीपल हु हैव टेकन इट दे हैव बीन सेव्ड. इफ by chance it is repeated, so we can save ourselves. So that's why it's better to take rather than not to take. So similarly, chloroquine and other drugs, they were widely used for the treatment of. Later on, it was assumed, even remdesivir, which was assumed that the most effective drug, later the DCGI told that it does not have any significant role for the treatment of the coronavirus. So still the work is going on to establish the permanent cure. Various companies have come up. They are preparing the protein-based these vaccines. They are always. So it's still going on. So CSIR in India, favipiravir, remdesivir, and colchicine are currently underway for the CSIR. They have made a combination. Central Council for the Scientific and Industrial Research Laboratory. They are working on to it, and they to develop to the particular medicine for the treatment of the coronavirus. Similarly, many other drugs which are being used for the influenza, flu, they are also being used, like favipiravir, and all these drugs are being used for the treatment of the COVID-19 patients. Next, एंटीपैरासिटिक. सबसे ज्यादा आइवरमेक्टिन का नाम सबको याद हो गया होगा. आई डोंट नो हाउ मेनी ऑफ यू. आइवरमेक्टिन जिसको दो एक आइवरमेक्टिन जरूर लिख दिया उस वक्त. It does not have any role. What is the basic role of ivermectin? It is antiparasitic drug, antiparasitic drug. पेट के हुकवर्म को मारने के लिए. कहीं कोई नहीं. अब कॉन्सेप्ट जहां से भी आया हो, किसी के पास कोई स्टडी डेटा नहीं है, बस एक होता था भेड़ चाल, क्योंकि हमें उम्मीद थी कि हम बच सकते हैं, तो हर ड्रग चली. तो उसमें सबसे ज्यादा आइवरमेक्टिन बिकी. That is basically, but however, it has also not been established so far that it was effective against the COVID-19 treatment. However, some of the people are claiming that they have been cured. So that's good if they have been cured. So that means there are chances. That's why it can also be considered as a drug for the repurposing. Tocilizumab, that is our interleukin-6 receptor that is working anti-inflammatory. That was also being used for the treatment of the COVID-19. Next. ये मेरे बारे में लिख के आ गया इसमें. बस मैं 5 से 7 मिनट और लूंगा. सर इजाजत है इतनी? हॉल भर गया भाई. चाय का टाइम हो गया क्या? Basically, I just wanted to show the table. 6 tables are there where I will tell you that this drug is being used for this purpose, but nowadays it is also being repurposed after carrying out all those screening which I was talking about, that the in silico, this work is going on. It does not been stopped. And very soon, many of the drugs will come out which are being used for the X disease are being sold for the treatment of Y disease as well. So this is the easiest part. Instead of investing the huge amount of money, the paradigm shift is there, that instead of the pharmaceutics only that they are changing the formulation, we can go for the change of the pharmacological activity. So that's why the role of the chemist, because we are just like, हाथ में तो 5 होते हैं लेकिन 4 हैं आपके बेसिक ओरिजिन, स्टैटिक्स, केमिस्ट्री, कोलॉजी एंड कोग्नोसी. We can, if we are individual, we can't do anything. We are like this. Whenever, until and unless we are together, I never say that the pharmaceutics is best or pharmacology is best or chemistry. When we are together, then only we can succeed. Ultimately, a drug which is being made by the chemist is useless until and unless the stable formulation is not developed. And if the stable formulation is not there, has been developed, but is having the numerous side effects, so the role of the pharmacologist is there. So that's why that is also integrated. And the fourth thing which we always neglect, that is the pharmacognosy, we cannot neglect. The origin of the chemistry, the structure or molecule is from the natural product. Initially, the first drug which were, still today, we have to keep an eye onto the animals. The forefathers, how they identified. When there was a disease, they keep an eye onto the animals. When they are having this kind of problem, which plant they are consuming. Based on that, they extract and they start consuming. So दादी माँ के नुस्खे जो कहलाता है वो कहाँ से आया? वो यहीं से develop हुआ. Basically, they observe and from there, by the observation, it is being used. So once the plant is not there, because they are not throughout the year, periodically. So what has to be done? The chemistry came into the existence. So first we have to extract out and find out the active ingredient present in it, and we have to synthesize in the laboratory when the season is not there. Biotechnology came into the existence. When the season is not there, we can develop that plant into the laboratory. So this is the development, and we are the part of. It does not mean we are in the develop. We are in the phase of the developing phase. And always, as long as you are in the developing phase, there is a chance, there are chances of the growth. When we feel like we have learned everything, that means our finish has been started. So the same way, the student, teacher, and everywhere, we have to keep on. कहीं से भी दिखा दो, बच्चों को बाद में पीपी दे देना, कोई नहीं, अब मेरे ख्याल में ज़्यादा बोल रहा हूँ मैं, तो बस कहीं से भी दिखा लो. जैसे sunitinib hai. This is a drug which is being used for the anti-cancer, imatinib resistant gastrointestinal tract cancer, and it is basically nowadays it is being used for the pancreatic neuroendocrine tumor for treatment. Similarly, for the tamoxifen, which is anti-cancer drug, which is used for the breast cancer, after the repurposing, it is being used for the systemic lupus erythematosus. That means the breakdown of the RBCs. That is, it is being used for the treatment of that also. Thalidomide, the well-known drug which has changed the concept of the stereochemistry. That was the first drug. I don't know how many of you are aware that is a thalidomide tragedy, R and S only. That means the orientation of the OH group, whether it is on the right-hand side or left-hand side. Only this kind of changes develop the teratogenicity. And because it was given for the motion sickness, and the lady who had consumed during their pregnancy, the kids newly born does not have the limbs. So this was a tragedy, and the drug was withdrawn from the market. And later on, after 20 years, the same drug has been relaunched for the different purpose with a different orientation. In place of R, that is now S. It is in the market form. So this way, the same drug is having the different kind of activity. So this is nowadays also used for the multiple myeloma, and the leprosy it is being used for the after repurposing, which is initially used for the motion sickness, ulti, uske liye to ab use ho rahi hai. Similarly, valproic, I'm just going the random way, valproic acid, which is used for the anticonvulsant drug. Now it is also being used for the manic disorder. Valsartan, antihypertensive. It is also being used for the AD. What is AD? Alzheimer's disease. Alzheimer's disease. That means it is now being used for the treatment of the Alzheimer, which was not thought of earlier. We have to develop new medicine, but now Alzheimer's is being treated by the same medicine. अच्छा, और तुमने और भी स्लाइड बहुत जल्दी भगाई, बहुत अच्छा किया. चलो ठीक है, आगे आ जाओ. इशारा उधर से हो रहा है? So, चलिए ठीक है. Similarly, there are plenty of the medicine which have already been either established or under the process. So, what are the opportunities? I will just only read out the first 5 points. Reduce the time and the cost. Drug repositioning is also low risk strategy because all the previous side effects have already been tested and it has been established medicine. That's why the low risk strategy is there. Computational or the machine learning approach, we have to learn more about the AI and the software so that we can develop the good molecule. Mixed approach in which the traditional as well as the newer one. As it is normally said in a house, why the old people are required, the parents and the grandparents, because they give their idea as the older one. And we have to incorporate, we cannot keep them out. The same way the mixed approach is the best approach. The older technique and the newer technique, they are mixed together, and based on that, we can develop a new molecule. So just like our families. R&D investment, we have to find out how much we can invest into the R&D because this cost is too high. And after the repurposing, we can reduce the R&D cost. Next. Challenges. The same thing which I talk about, that this is the advanced technology we have to learn. We have to establish the commercial model, means whatever we are doing, we should find out a collaborator who can carry out the screening for ourselves, because one individual cannot carry out each and everything. Even if one industry cannot do, that's why the interdisciplinary research is also coming up very faster. The same way the pharmaceutics, pharmacology and pharmacochemistry, this is also a kind of interdisciplinary approach altogether. When they become like this, it behave like this. Individually, we can't change the anything system. Patents, we have to go for the patents. This is also a kind of difficulty also. The drug which has already been patented for some disease and it has been established by myself, how can I allow this medicine to use for the other person? This is the important challenge that has to be looked into. So that's why the most of the repurposing concept is being applicable for those molecules which have already, in which the patent is already expired. If the patent is already there, so we have to take the permission from the person who has obtained the patent, and then only we can go for the repurposing. This is a major side effect of the repurposing of the drug. So either we have to wait the expiry of the patent, or we have to collaborate with the person who has developed and who had obtained the patent for that particular medicine, not for that particular disease, but the molecule has been patented for him, so he can get the revenue of that. So this way we can have to develop the next. Regulatory approval, I just told you, IPR is the most important thing where the regulatory is there. So regulatory agencies have to come forward and look into how can we facilitate for the repurposing so that the interest of the humankind rather than the industrially has to be minimized. As of now, definitely if I am investing, I am developing a molecule, so it is my privilege that I should get the maximum amount. But at the same time, not the cost of the life of a person. So that's why we have to establish the IPR and the DCI office. They have to come forward. So how can we minimize these effects so that the repurposed drug can be launched in the market? Because many of the time it has been observed that the drug has about to be launched, but the patent issue has come forward and that could not be launched into the market. So that's why majorly the work is going on for those medicines in which the patent has already been expired. बस यही बात कहनी थी, बस जल्दी से यही हो गया कंक्लूजन. चलिए यही बात हो गई. चलिए recommendation. There are several recommendations which I have already recommended, although there is no DCGI office person present. But I recommend that we have to keep in mind for the human life rather than the industry. I must recommend that they can dilute the time period for the IP so that the people who can be benefited for the that particular medicine, the life is more important than the money. This is my submission to all. And if there is any question related to this, you are welcome to ask. हाँ वो तो मजबूरी है खैर. जी, if there is any question, आप लोग बोले, मैं ही बोलता रहूंगा, वरना सही बात है. आदत बहुत खराब है, बहुत खराब टीचर हूँ. सुखेंद्र अब कभी नहीं बुलाएगा मुझे. मत बुलाओ, नुकसान किसका है ना, तुम न बुलाने की.
If anybody have a query, they can ask.
ये बॉटल भी किसी की पी हुई रखी थी यहाँ पर। उतर जाऊं? Any question?
Thank you so much, sir, for your valuable information.
तो हर बार ये कहूंगा तो हर बार ताली बजेगी या उतरने के लिए? थैंक यू वेरी मच।
Excuse me, sir. I would request Professor Dr. Garima Garg, Dean, MIT College, and Professor Dr. Arun Kumar, Principal, Parmanath College of Pharmacy, to facilitate Dr. Shahar Yar Khan.
It is an honor to me to receive the honor from my ex-colleague. We have worked together in MIT from 2000 to 2003. We worked together.
Honor to me. I got the opportunity.
तो कहते जी आप भी आइए हमारे लिए, हमारा honor और करें।
Thank you so much. With Professor Dr. Garima Garg, Dean, MIT, and Professor Dr. Arun Kumar, Principal, Parimarth College of Pharmacy, as the chairperson and co-chairperson, I'd like to invite Ms. Nidhi Dhama to introduce Dr. Sushil Mishra, Senior Research Scientist Group Lead, Analytical Research Department, Integral Biosciences Private Limited, Noida. Please, ma'am.
Thank you, dear students. Jai Hind and good afternoon to all. It is a great pleasure for me to introduce our speaker, Dr. Sushil Mishra, Senior Research Scientist and Group Leader, Analytical Research Department at Integral Bioscience Private Limited, Noida, which is a contract research organization with specialized expertise in early stage contract research, medchem synthesis and development, analytical development, and small molecule analysis and purification. Dr. Sushil Mishra, sir, completed his B Pharm and M Pharm from University of Delhi with wonderful grades in 2004 and 2006. He also qualified GATE exam in 2004 with 92.3%. Sir completed his doctorate from Delhi University on development of intelligent polymeric formulation for enhancement of bioefficacy in Department of Quality Assurance. Dr. Sushil Mishra has expertise in structure elucidation and characterization of new chemical entities and in LCMS method development for medchem QC checks and parallel medchem libraries, including mass tuning, product ion, and precursor ion MS scan. Sir also has wonderful skill in data interpretation and report generation for homonuclear and heteronuclear 2D NMR experiments like HMBC, HSQC, COSY, and NOSY. Sir also has ability to work with external customers from the global pharmaceutical and non-pharmaceutical industries. He also has excellent communication and customer interaction skill. He has ability to provide a strategic direction to a large analytical team with the necessary technical leadership. Sir also has so many publications in Scopus Index, Web of Science Index, and Elsevier Index journals. Sir also delivered so many lectures in many universities. So here I am requesting to Dr. Sushil Mishra, sir, please come and explore the knowledge about the opportunities and challenges in pharma sector.
Thank you so much.
Thank you.
So, good evening, Jai Hind, and good evening to all of you. So, I'm Sushil Mishra, and today's my topic of seminar is to introduce you what is the analytical method development and validation. So, जस्ट एक छोटी सी भूमिका के साथ मैं शुरू करूंगा कि सपोज मैं आपको एक पैरासिटामोल की टैबलेट देता हूं और आपको बोलता हूं कि आप इसे एनालाइज करके मुझे बताइए, तो आप क्या करेंगे? आपका आंसर होगा कि सर हम फार्माकोपिया ओपन करेंगे, फार्माकोपिया में जो पैरासिटामोल का चैप्टर है, जो मोनोग्राफ है, उसे देखेंगे और जो भी टेस्ट उसके अंदर प्रेस्क्राइब्ड हैं लिमिट के साथ, स्पेसिफिकेशन के साथ परफॉर्म करेंगे. I will ask, do you have a confidence on this result? You will say definitely yes. Why? Because ये validated है. Nobody can challenge. Wherever you will perform this test in any country, if the situation is same, it will deliver the same results. So this is the power of validation. फार्माकोपिया के अंदर जो भी मेथड, जो भी स्पेसिफिकेशन या जो भी टेस्ट हैं, वो कहीं ना कहीं से वैलिडेट होकर के ही आए हैं उसके अंदर. So basically validation is next. So basically validation is defining or establishing a documented evidence which provide a high degree of assurance that specific method which will you use will consistently perform and give the same results. Next. So the objective of any validation parameter is to have a consistent, reliable and accurate data. Consistent means आप उसको end time of perform कीजिए, end time of analysis कीजिए, हमेशा result वही के वही आएंगे, ठीक है? And they are reliable because you are confident enough to report it, and they are very accurate data. Nobody can challenge. Next. So why analytical validation is required and what is its important? Let us take another example. हमने एक स्पोर्ट्स इवेंट को ऑर्गेनाइज किया, इंटरनेशनल और नेशनल प्लेयर्स आए हुए थे उसमें, और हमें उसमें डोप टेस्ट करना है. एंड सम ऑफ द प्लेयर वर फाउंड कि दे वर टेकिंग सम काइंड ऑफ द ड्रग, दे विल बी बैंड. They may go to the court and challenge you that you have spoiled their career. In that case, you have a valid method, validated method. On the basis of that, you can challenge them. So this is the power of a validated method. Number two, establishing a new method of the analysis over the existing method. It means that if you see the pharmacopoeia, you will find some of the assay methods, especially of the APIs, they are by the titrimetry methods, like non-aqueous or potentiometry method. Now you have developed a better or superior method. Now there are two methods available. Which one is true? So in that case, you have to perform the validation of that method. Thirdly, in case of method transfers, nowadays what happens? If you see example, let us take an example, Sun Pharma. Sun Pharma ka R&D is in Gurgaon, but their plant is somewhere else. So R&D scientists will develop a method and transfer to the QC scientist of the plant. So this process required a method validation procedure. Fourthly, demonstrating equivalence between the two methods. Let us take another example कि मेरे पास एक HPLC method है और मैंने एक superior method UPLC बना दिया. Both are valid, but दोनों को validate तो मुझे करना ही पड़ेगा. Fifth thing is there is a regulatory requirement also. Whenever you are going to the regulatory authorities for NDA or ANDA filing, they will definitely ask for the method. In that case, you have to give a validated method protocol. Okay, to begin with, there are 6M or some prerequisite things in the analytical method development. These are the men. Okay, sorry. So we need a man, machine, method, material, milieu, and management. So what is a man? Man is the manpower. You all are the manpowers. But you need a qualification and you need a skill to perform the whole activity. Machines, you are ready, and then what we need? Machines, HPLC, UPLC, or whatever. So these machines must be calibrated, they must be qualified, and they must be robust enough to give the results. All these things are ready. What else you need? Method, obviously. The method should be properly characterized, it should be stable, and it is properly well documented. Then what you need? Material. Material what? It's a reference standard or a certified reference standard. Then only you can perform the validation. Next is the environment, lab environment you need, that has to be 25-degree humidity control, no electrical noise or no vibrations, like. And finally, you need the management, time management, resource management, chemical management. So when all the 3 M's are complete, then only you will approach for a validation of a method. Next. So this slide depicts what is the life cycle of the analytical method. Analytical method, sorry. So we start with the planning. So in the planning, we first define what is the goal and what is the description and what is the objective of method development. Once it is defined, you do some kind of literature search, whatever the literature search is available or not available. So on the basis of that, you compile everything and approach the method development. So in the method development is the first step, initial step, or the baby step in the lab, where you check the stability, the purity of the material, and you develop the methods. Then this method is further optimized in every region, in every aspects. Finally, it is given to the R&D scientist. They check it rigorously, and when all things are okay, fine, and everything you are getting good, then you will go for the validation experiments. After that, the method is transferred in case of the QC department, or the method could go for the filing to FDA or pharmacopoeia. All things are complete. Then finally what? You need to periodically monitor or review the method continuously, so that if there is a new requirement, you must be ready for that. Next. The question comes, where is the analytical validation is performed? It is performed in GLP or by the QC scientists. What is the source of the standard material? It is important to note that standard material should be some certified reference material, or it should be a working standard at least. Next. Who will perform the analytical validation? Yes, you can perform, but you need a GLP certification, training, and proper skill. Next. So next slide says, what is the difference between the verification and validation? So there is a small example. Suppose you are a contract manufacturing organization. You have received order from a patent person or from a license holder that you have to produce or manufacture some drugs, formulations. So what you will do? You need a method for the QC purpose. So there are 2 choices. Next. So there are we have 2 choices. Either you will develop in-house method, or you will use the compendial method or pharmacopoeial method. Next. So when you will develop your method, and under what condition you can use a compendial method. So I have taken example of diclofenac sodium. So diclofenac sodium, if you can find in pharmacopoeia, it is registered as an API, tablet, creams, and injections. So if these formulations are there and already pharmacopoeia is giving you the method, in that case, in your laboratory or in your QC department, you have to verify it. No need to perform the validation. But in case these formulations are not registered and you have any nano formulation, any delayed or sustained release formulation, you have developed any patch or any other novel delivery system which is beyond the pharmacopoeia right now. So in that case, you have to perform the method validation of the process. Next. Then a word comes, revalidation. Verification, you understand. And now what is a revalidation? So revalidation is a process that you have already validated a method by a series of experiments. Now suddenly what happens, your synthetic route is changed, your formulation route is changed, or you change some kind of the excipients, in that condition you need to revalidate. Or suppose you are using an X column, now you are using some other brand column. So in that case, you have to revalidate the method. So the revalidation is not necessary to complete the whole parameter, only the change parameter, whatever changing you are bringing and whatever it will affect the further has to be validated. What is qualification? Suppose there is some job advertisement. So job advertisement says that we need a qualified M.Pharm or B.Pharm person. The question comes, how we become the qualified person? You have studied 4-year or 2-year program. You have given the exams. You have passed. A competent authority on the basis of your performance results give you a passing certificate and a degree. Then only you become a qualified person. It's okay for the manpower, but what happens to the machines? Machines do not take part in any school. They don't go to universities. So the machine needs a qualification. And this qualification is what? They have to undergo some kind of the test. These are the calibration tests. Whatever movable parts are there, especially the movable parts, then they will be calibrated. And once they are calibrated, documented, and you are getting good results, you're confident enough, then it is said to be qualified. So this is the qualification parameter of a machine. So what is the difference between next? Okay, sorry, bhul ja raha hu. So what is the difference between qualification and validation? So validation and qualification are essentially component of the same concept. Qualification is normally used for the equipment, premises, utilities, system, column, etc. But validation is only used for the process and method. Finally, I will say a process validation cannot take place or cannot be initiated if qualification is not done. Next. So finally, we have come to the analytical validation. So what are the analytical validation to be performed, or what are the guidelines? So these guidelines are available online. So you can check with the ICH, you can check with the FDA, respective pharmacopoeias or European guidelines. Next. So these are the 9 parameters or attributes which will be covered for the method validation: specificity, linearity, range, accuracy, precision, detection limit, quantitation limit, robustness, and system suitability parameter. Next. Okay, this chart explains under what condition and which type of the validation parameter you need to cover. So first column is for the characteristic Okay, next slide. Next. Okay, the first topic or the first step is the specificity. Specificity means what? आपने एक method validate करना है. तो first thing you should know कि that you have a correct API. आपने incorrect API choose कर लिया तो फिर तो आगे blunder ही हो जाएगा. आपके पास सफेद पाउडर पड़ा हुआ, it can be paracetamol, diclofenac, anything, any white drug it can be. So you need to first specify that whatever material you are having, you have to be confident enough that is the target that you have to achieve. So this is the specificity. Specificity is performed in number of ways. First is the identification. Identification means that you have to establish only the identification protocol, nothing else. If you'll see in the pharmacopoeia, there are certain identification tests. It may be IR, it may be NMR, it could be mass, or it could be DSC, XRD in case of any kind of crystalline material is there or amorphous material is there, right? So the purpose of all these tests is to just identify, need not to quantify. Next. Okay, the second thing is you have to develop some specificity for the assay or impurity test. So guideline says to use the known impurities. Next. Okay, there is something missing here. Okay, so what you can do is you can spike the unknown, you can spike the impurities along with your API, and you can check the resolution. And on the basis of that, you can discriminatingly say that the target is present specifically. Next. If impurities are not available, why? Because impurities are very costly. Just 2 to 3 milligram can cost 2 to 5 lakhs. So generally impurities are not present. So in that case, what to do? Next. The guideline says that if impurities are not present, you can do the stress stability studies. These are the stress stability parameters that you can degrade your molecule into acid, base, peroxide, heat, light, humidity. The purpose is to generate forcefully some impurities. And in the presence of that impurities, you have to identify, specify your API. Next. There may be a case where the API is not stable. You plan some kind of degradation and API is completely finished, gone. Nothing is there. Only impurities are left. So what to do? So in that case, you can perform the peak purity determination. Peak purity determination, for that you need a diode array detector or PD detector, HPLC. Right? Then in that case, you will plot a graph. So in the first graph where you can see the peak purity, there is a blue line and a green line. So if they are not intersecting each other, it means that the purity is passing. It means that the peak is having no collision of any other unknown or known impurities. Whereas in the second graph, you can see something is there in the tailing. So the peak purity is failing. So there is some kind of illusion, right? Co-illusion, sorry. Next. Okay, next. This is the important one, the linearity. So what is linearity? Linearity of an analytical procedure is its ability to obtain a result which are directly proportional to the concentration. For establishing a linearity, you minimum have to have a 5 concentration. Next. So this is a linearity curve. There are certain components which I will elaborate. You can see the R square. This is the correlation coefficient. It has to be maintained a very high value, more than 0.99. Then you will find an equation, y is equal to something plus 100, 0.019. So this is a slope equation. So y signifies the slope of the regression line, whereas 0.019 signifies the y-intercept. So what is the significance of R square? R square says that whatever you will do the calculation, whatever the response is there, it is completely linear to your concentration. So if it is 0.99 or it is approaching to the perfectly 1, it means that your Beard-Lambert law is validated to a certain range. Next. So this range is defined by the guidelines that upper and lower range you have to select. And for that, if it is an assay method, it has to be 80 to 120%. If it is a content uniformity, it has to be 70 to 130%. Next. This is a very good thing. Accuracy. Okay, so you are performing some kind of a test, like paracetamol का आपने कोई method develop किया, claim is 500 milligram, but each and every time of the analysis you are just getting a 400 milligram recovery. Each and every time. That means accuracy is failing. So you can see the bullseye. So your true value and your mean value should be very close enough to prove the accuracy. Next. Okay, next. Directly come to the precision. Come to the next. Yes. What is precision? Precision is the expression of the closeness of the agreement between the series of measurement obtained from the single sampling of this homogeneous sample. Next. So this picture depicts four things. The first diagram says that your results are accurate but imprecise because they are far away from the mean value, the bullseye. The second diagram says where all the arrow are concentrated at a single position, though they are precise, but they are inaccurate because it is still far away from the true value. If you see the third one, all values are scattered, all results are scattered, all arrows are scattered. So it is inaccurate as well as imprecise analysis. Whereas the last one, where you will find all arrows are hit on the bullseye. So you can say that your method is accurate and precise. So accuracy and precision go hand in hand. Next. So precision can be performed in a three-way: repeatability, intermediate precision, and reproducibility. So repeatability is the intraday precision, that you have to perform this analysis in a single day within a limited period of time, 1 hour, 2 hour, jitna jaldi ho sake. Intermediate precision, it is performed over the period of time, say first day, second day, third day, fourth day, like that. Reproducibility, it is a comparison of the result produced in one lab and compared with some other lab which is at some other premises. Next. Next table aayegi. This is the table. So this signifies what test to be done in case of repeatability, intermediate precision, and reproducibility. So I have compared some of the points, like in case of laboratory, in repeatability precision, the lab has to be same. In intermediate precision, the lab has to be same. But in case of reproducibility, definitely the premises and lab has to be different. Second point is analyst. In repeatability, the analyst is same, whereas in intermediate precision, different analyst has to be there. 2 or 3 different analysts will perform, and then they will compare the result, so that there is no error due to the manpower or any kind of analysis, right? So in reproducibility also, there will be the analysis will be different. Coming to the apparatus or machine or the column or the chromatography system, the apparatus will be same in the repeatability test, whereas in precision also it will be same, whereas it will be completely different in the reproducibility. The timeline, as I said, in case of repeatability, it has to be short, the same day, maybe 1 hour, 2 hour, or 3 hours, something like that. Don't go beyond that. Whereas in case of intermediate precision, it is a multiple day, 1, 2, 3 days, 4 days. जब तक आपका solution, आपका analyte stable हो सकता है, तब तक हम उसको करते हैं, ठीक है? And reproducibility, definitely nothing is specified, so no need to perform. Next, this is a typical table where I have shown what are the data requirement or stats requirement in the precision. You need to have an average value, you need to have a standard value, and percentage RSD, which should be less than 2%. Next. So all things are there, then you can say that your results are precise. This comes the LOD and LOQ. So the word itself says there is a limit of detection and there is limit of quantitation. That means that this is the lower limit which can be detected but not to be quantified. This comes where whenever you are developing some kind of the limit test, in pharmacopoeia, you will find some kind of the limit test, chloride, sulfate, or something like that. In that case, you have some standard solution and you compare the opacity. So for that purpose, you need the detection limit, right? And when it comes to the quantitation, especially the impurity profiling, for that purpose, impurity profiling purpose, you need to have a limit of quantification. That means how much the low impurity your method can quantify. Next. Yes. So one of the parameter of finding out LOD or LOQ is the signal to noise ratio. You can see the noise level is there. So 3 times of the noise is LOD and 10 times of the noise is LOQ. Next. Robustness. Robustness means if there is any kind of deliberate changes in your method due to the machine, due to the column, pH, mobile phase, or analyst, your result should not be affected. Or if it is getting affected, you have to properly document that these are the specifications, these are do's or don't do's, under which the whole operation will be performed. Next. Okay, next. Yeah, this is a table you can find. This is a robustness. There are 2 parameters are being checked, a buffer and buffer concentration. So if you see the buffer, there is a deliberate change in the buffer, 5.9 and 6.9, but there is no change in the values. Whereas if you change the buffer composition, there is a drastic change. So that means that your method is robust enough to grasp or to feel the change in the buffer, or your buffer capacity is good enough to control any kind of the change, but your mobile phase composition is not good enough. If you change all the values will drastically change. So in that thing you have to document it, and that thing has to be reported. Next. System suitability tests. Okay, once your method is developed, validated, and it is documented, and it is transferred to the QC. So every time they will not do the validation protocol. So what they will do, they need some system suitability test, which will check the performance of machine, column, and your material. And if all these things are passed, then only you will start the analysis. So normally what happens, the API or the formulation is spiked with some known impurities or any other drug, and the resolution, the peak shape, symmetry factor, capacity factor, and resolution are checked. They are defined as a specification, and once they are qualified, they are okay, then only you will perform your test. Thank you so much. So I am very much thankful to the organizing committee, dean, faculty of the pharmacy, and pharmacy, all the faculty members. Next. So something about the IBS. IBS is a discovery CRO company situated in Noida. Our prime goal is to find out the hit drug molecules. So we are designing the drug and we are finding out the target of the drug. So this is a company where the drug is not, this is not the drug actually. This is the new chemical entities. They are not even the drug, because once a chemical is registered, then only it becomes the drug. So these are not the registered one. So we extend our analytical support help to all the PGs, PhDs, and other research scholars. You can contact us for any kind of analysis help. We do offer some AMFORM project trainees. So we are having almost every year someone from the DIPSAR, Galgotia, Amity, APG, and Lovely Professional University. So students are coming, AMFORM students are coming for their project work. Next. So this is something which I want to show. This is the scientific life or the scientific research life at IBS. This is my lab. People are working on HPLC, UPLC, mass. Next. So, okay, the first picture is about the auto purification system. So there is a HPLC and there is a bada wala HPLC. Bada wala HPLC क्या करता है? वो purify करता है. That is a preparative machine. So suppose you have a molecule you need to be purify or you need to isolate an impurity. So this machine does all the work. So second picture, the middle one is the SFC, supercritical fluid chromatography. It is a more advanced type of the liquid chromatography where it uses the liquid carbon dioxide as the mobile phase. Once the carbon dioxide is liquefied, it undergoes the supercritical phase and it behaves like a solvent. The last one is for our 400 megahertz NMR. Next. So this is my bioanalytical lab. So we have LC, MSMS, and you can find the people and scientists are working in that lab. Next. So this is the final slide. And this is from our synthetic chemistry lab. So scientists are working. And in the synthetic chemistry lab, to follow some protocol, you have to wear the lab coat, you have to wear the mask, you have to wear the gloves and goggles. Every safety parameter has to be followed. And these things you need to cultivate right from the beginning when you are in a college, right? Because safety is the first thing. And whenever you are dealing some kind of experiment, something unsafe can be there, could happen, an accident could be there. So this may be a life threatening. So always prefer and always take safety gears before starting any experiments. Thank you to all of you. Thank you, sir. So if you have any question, I don't think time is there.
Thank you so much, sir, for the wonderful session. The question answer session is open. If anyone want to ask any question, they can ask.
Thank you so much, everyone. Now I'd like to invite our chairperson and co-chairperson, Professor Dr. Garima Garg and Professor Dr. Arun Kumar to felicitate Dr. Sushil Mishra.
Thank you so much.
Thank you so much, everyone. Now I'd like to invite Dr. Ganesh Mishra, HOD, Department of Pharmaceutical Chemistry, Professor Dr. Sukhinder Kumar, Dean, Faculty of Pharmacy, and Mr. Radesh Kumar, Assistant Professor, Faculty of Pharmacy, to felicitate our chairperson and co-chairperson for today, Professor Dr. Garima Garg, Dean, MIT, and Professor Dr. Arun Kumar, Principal, Parimarth College of Pharmacy. Please give a huge round of applause.
Huge round of applause, everybody.
I'd like to invite Ms. Nidhi Dhama, Assistant Professor, Faculty of Pharmacy. Please, a huge round of applause, guys. Now I'd like to invite Dr. Ganesh Mishra to welcome Dr. Supriya Mehti, Principal RSD Academy College of Pharmacy, Muradabad. Dr. Amit Punia, Assistant Professor, Faculty of Pharmacy, will be receiving the memento and Angvastram on behalf of her.
Give a big round of applause, everybody. Now, I would like to invite Mr. Prasenjit Paul, Head of Department, Pharmaceutics, Faculty of Pharmacy, to welcome Dr. Ratindra Kumar, Principal, Neelkanth College of Pharmacy, and on the behalf of them, Ms. Shiva Rajput, Assistant Professor, will be taking the Angvastra and mementos. A huge round of applause, everybody. Thank you so much, ma'am. Now again, I would like to invite Mr. Prasenjit Paul, co-convener, to summarize the conference.
Jai Hind, everybody. I am presenting before you the brief summary of today's one-day national conference entitled Opportunities and Challenges in Pharmaceutical Sector with the theme Unlocking the Potential to Solve Challenges, organized by the Faculty of Pharmacy, Swami Vivekananda Subharti University with the Association of Indian Pharmaceutical Graduate Association and IQAC, SVSU. The conference initiated with the lamp lighting ceremony and Saraswati Vandana. This was followed by distribution of sapling to all the eminent dignitaries present on the dais. The souvenir of the conference was released in the presence of Honorable Vice Chancellor, Sir, Guest of Honors, Professor Arun Garg, Dr. Neetu Pawar, IQAC Director, and Professor Dr. Sokinder Dean, Faculty of Pharmacy, with the assistance of Associate Professor Mr. Manish Kumar Pathak, the organizing secretary of the event. The inaugural session began with a welcome address by the convener of the conference, Professor Sokinder Kumar. In his opening remarks, he extended his warm welcome to the Vice Chancellor, guest of honors, eminent speakers, and deans of other colleges, and heartily appreciated their presence in the conference. He also welcomed the faculty and student delegates from different colleges and participants of the poster presentation for being a part of the conference. The event was briefed by Professor Dr. Ganesh Prasad Mishra, co-convener of the conference, highlighting about the objectives and motives behind the conduction of the conference. He also introduced the theme of the national conference to all the participants of the conference. Thereafter, session was further graced by Honorable Vice Chancellor Sir of Swami Vivekananda Subharti University, Major General Dr. G.K. Thapliyal. He firstly acknowledged all the eminent speakers and panelists who have joined today's conference. He congratulated the selection of today's theme in the conference. He mentioned various challenges in medical and pharmaceutical sector, mentioning the acute shortage of medicines, including PPE kits, especially during COVID pandemics. He also emphasizes importance of the growth of medical tourism in India. In his address, he stressed upon the learning the ways to imbibe advanced technology in the field of pharmacy, particularly by collaborating with the foreign countries. After the address of the Vice Chancellor, Sir, Ms. Nandini Sharma, Assistant Professor, introduced the first guest of honor, Professor Dr. Arun Garg. Honorable Vice Chancellor, MVN University, Palwal, Professor Arun Garg is in his address stressed the importance of becoming self-sufficient in various fields, especially medical and pharmaceutical, which will boost the national economy and decrease the country's import bill. He also acknowledged the speech of Honorable Vice Chancellor Sir of Subharti University. He advised the student to incorporate the teachings of Swami Vivekananda in our daily life and become an ideal citizen. Finally, he was felicitated by Honorable Vice Chancellor Sir. The session carried on with the welcome of second guest of the honor, Professor Vibhu Sahni, Department of Pharmacy, LLRM Medical College, Meerut, by Ms. Swati Vadhavan, Assistant Professor. In his address, Professor Sahni discussed the future roadmap of PCI in framing the policies of pharmacy courses. He discussed the reforms in pharmacy, educating the introduction of exit exam, online registration process, framing industry-based syllabus, creating pharma portal, a job portal, and the concept of healing by Indian at foreign countries. He was felicitated by Dr. Manish Kumar Phatak, the organizing secretary of the event. Thereafter, the first scientific session pre-lunch occurred. The first scientific session was started under the chairmanship of Professor Dr. Sachin Tyagi, Principal, BIT Meerut, and co-chairmanship of Dr. Sachin Kumar, Principal, NKBR, Meerut. After that, Dr. Kunal Arora, Assistant Professor, introduced the first speaker of the session, Professor Vijay Bhalla, Dr. Vijay Bhalla, Principal, SGT College of Pharmacy, SGT University, Gurugram, Haryana. Professor Bhalla, in his address, gave a nice presentation on today's theme of the conference. In this, he advised the student to be well aware of the recent modifications of Drugs and Cosmetics Act and also Pharmacy Act, and encouraged them to become a self-entrepreneur, and told them the importance of forming brand. After his address, he was felicitated by chairman of the session. This was further proceeded by the introduction of Dr. Jai Prakash, who is the Senior Principal Scientific Officer, Indian Pharmacopoeia Commission, Ghaziabad. By Dr. Charu Saxena, Assistant Professor. Dr. Jai Prakash, in his address, gave the basics and overview of pharmacovigilance program in India. He described various activities relating to the understanding and prevention of adverse effects of drugs and pharmaceuticals, including side effects of vaccines. He also encouraged students to undergo their research work at IPC Ghaziabad. Later, he was felicitated by chairman and co-chairman of the session. At the end of the session, first scientific session, the convener felicitate both the chairman and the co-chairman. Post-lunch, the second scientific session began. The session started under the chairmanship of Professor Dr. Garima Garg, Dean, MIT Department of Pharmaceutical Technology, and co-chairmanship of Professor Dr. Arun Kumar, Principal, Pharma College of Pharmacy, Meerut, both of whom were greeted by the convener, Professor Dr. Sokindra Kumar. The first speaker of the session, Professor Mohammad Shahar Yar Khan, Department of Pharmaceutical Chemistry, Jamia Hamdard, spoke about the potential to develop new molecular entities warranting clinical use in human beings and animals. He also differentiates the traditional drug discovery versus drug repurposing, including strategies and methods of repurposing. He also motivated to increase the use of artificial intelligence in the field of drug discovery. After the address, Dr. Shahar Yar Khan was facilitated by chairman and co-chairman of the session. The event progressed with the introduction of Dr. Sushil Mishra, Senior Research Scientist, Group Leader, Analytical Research Development, Integral Biosciences Private Limited, Noida, by Ms. Nidhi Dhama, Assistant Professor. He gave a presentation on analytical method development and validation. He discussed the importance of validation in the pharmaceutical industry. He was later felicitated by the chairman and the co-chairman of the session. Finally, both the chairman and co-chairman were felicitated by the convener. At the end, I am happy to announce that about 620 delegates from about 7 states of India, namely Uttarakhand, Uttar Pradesh, Haryana, Bihar, New Delhi, Rajasthan, Jharkhand, have participated in the conference, and about 55 posters have been submitted by different participants in poster presentation competition at this one-day national conference. With the support and steerage of different committee members, panelists, and amazing displays of the participants, especially of poster presentation, made this event outstanding. The theme of the conference, Opportunities and Challenges in Pharmaceutical Sector, with the theme Unlocking the Potential to Solve Challenges, was a great success, where eminent keynote speakers from various pharmaceutical disciplines made their respective presence and addressed the gathering. The national conference witnessed an amalgamation of peerless speakers who enlightened the crowd with their knowledge on various topics related to the field of pharmaceutical sciences. Thank you.
Thank you so much, sir. Now here comes the time for the prize distribution of poster presentation. For the same, I would like to invite Ms. Swasti Arora, Assistant Professor, and Ms. Swati Vadhavan, Assistant Professor, Faculty of Pharmacy, for the prize distribution.
Thank you, dear students.
Thank you, dear students, for inviting us.
Jai
Jai
Hind
Hind to all. We would like to invite our principal, sir, Dr. Sukendra Kumar, for giving prize to our deserving students.
Welcome, sir.
Today, we all have learned and enjoyed a lot from this conference, NCOCPS 2023. Wow, what an amazing posters presented by our students and respected delegates.
It was obviously a difficult task for jury to choose the winners, but our honorable juries did it so well.
Now, moving towards the award ceremony of poster presentation, we would like to announce the winners.
The criteria for the poster presentation is presentation, designing, content, and novelty. We have selected winners on the basis of abstract presentation, designing, and overall presentations.
Now, going towards the result, For abstract category, third prize goes to Arjun of B Farm second year. I would like to invite Dr. Manish Kumar Pathak sir to come and give the prize to the students. Students, please clap. A huge round of applause for the winning students.
Second prize goes to Piazza from Farm D first year KSCP. Dr. Kunal Arora sir, please come on the stage for the prize distribution.
First prize goes to Bobby and Shiv Kumar and team RSD Academy, College of Pharmacy, Muradabad. Dr. Amit Kumar sir, please come for prize distribution. RSD Academy team.
Next है presentation है ना? Second round is.
बहुत एनर्जी से इन्होंने पोस्टर बनाए हैं, क्लैप करते रहिए सब.
अब नेक्स्ट डायलॉग है ना, क्लैपिंग दिखनी नहीं चाहिए. तुम्हारा है अब दोबारा से, चलो.
Coming toward the presentation, for presentation category, there is a tie between Ananya Dev of B Farm second year and Pranav, B Farm first year, Kharwal Subharti College of Pharmacy. I would like to invite Ms. Dimple ma'am. Please come for prize distribution.
क्या बोलूं? तालियां बजती रहनी चाहिए. तालियां रुकनी नहीं चाहिए, प्लीज. Second prize goes to Sonam Tyagi from Ethnopharmacology, Dentistry, SVSU. Miss Nidhi Dhama ma'am, please come on the stage for the prize distribution. विशाल को तुम बुला लो ना, देखो.
सोनम त्यागी, डेंटल कॉलेज.
कोई और हो इनके बदले जो उनका प्राइज ले सके.
और कोई आएगा सोनम त्यागी का प्राइज लेने के लिए? First prize goes to Mayank Tyagi, M Farm, Kharwal Subharti College of Pharmacy. बेटा तालियां बजाते रहिए. टीचर्स भी प्लीज तालियां बजाते रहिए. Next is designing. For designing category, third prize goes to Sunny Vishal of B Farm first year, Kharwal Subharti College of Pharmacy. Vishal sir, please come for prize distribution.
Second prize goes to Amit Kumar Cafe Ansari, B Farm fourth year, KSCP. Mr. Adesh Kumar, sir, please come on the stage for the prize distribution. Need a number.
All are requested to please clap. For first prize, there is a tie between Anjali Kumari and team Accurate College of Pharmacy and Abhinav Kumar Singh, B Farm fourth year. I would like to request Ms. Swati ma'am for prize distribution. Last but not the least, the final category is overall poster presentation. In this category, third prize goes to Aman Kumar, Vishal Kumar, Diploma first year, Kharwal Subharti College of Pharmacy. I would like to call Deepak sir for prize distribution. Dharmen sir, please come for prize distribution.
For second position, there is a tie between Ritik
Vibha and Lakhan Vikrant. PharmD second year.
Dr. Vikrant Verma, sir, please come on the stage for the prize distribution.
Prashant sir, Nishant sir, please come on the stage.
लास्ट वाले में धर्मेंद्र को बुला देंगे.
Prashant sir, Nishant sir, please come on the stage.
first prize goes to Shivani Agrawal, Faculty of Law, SVSU. Dharmendra sir and Swasti Arora ma'am, please come on the stage for the prize distribution. कोई और उनके behalf में ना?
A huge round of applause for all the winners. Congratulations to all the participants, winners, and their mentors. Thank you.
Thank you.
Thank you so much, everybody, and congratulations to students. Now, I would like to invite Dr. Manish Pathak, Organizing Secretary, for a vote of thanks of this conference.
Jai Hind to all. Thank you. Such a prayer that cannot be seen or touched. It can must be felt by the heart. I feel honored and privileged to get the opportunity to propose vote of thanks on this special occasion. सर्वप्रथम मैं हमारी सबकी प्रेरणा स्रोत श्रद्धेय स्वर्गीय संगमाता डॉक्टर मुक्ति भटनागर को नमन करता हूं. And then thanks to honorable founder sir, Dr. Atul Krishna, for giving us an opportunity and blessing to organize national conference. Thank you, sir. I, on behalf of organizing committee, convey deep regard and heartily thanks to our patron, Honorable Vice Chancellor, Major General Dr. G.K. Tapalyal, sir, and Honorable Chief Executive Officer, ma'am, Dr. Professor Salya Raji. I would like to thank all the honorable delegates who blessed us. I would like to thank our guest of honor, Dr. Vibhu Sahani, Professor and Head, Department of Pharmacy, LLRM Medical College, Meerut, Professor Dr. Arun Garg, Vice Chancellor, MVN University, Palwal, Haryana, for their gracious presence. I would like to thank our resource person, Dr. Jay Prakash ji, Senior Principal Scientific Officer, Indian Pharmacopoeia Commission, Professor Dr. Vijay Bhalla, Principal, SGT College of Pharmacy, SGT University, Professor Dr. Shahar Yar Khan, Department of Pharmaceutical Chemistry, Jamia Hamdard, and Dr. Sushil Mishra, Senior Research Scientist, Integral Bioscience Private Limited, for enlightening us with the knowledge. I am thankful to the Registrar Sri D.K. Saxena ji for helping us in obtaining administrative approval for organizing this event. My thanks to Pro Vice Chancellor sir, Dr. Abhay Shankar Gora sir, for his blessings. My special thanks to Dr. Nitu Pawar, Director IQAC, for their support and guidance. My sincere thanks to all deans, principal, HODs for their presence in conference. I would like to thank our beloved Dean and Principal, Professor Dr. Sukhinder Kumar, convener of the conference, for his help and support at various stages that radiate a source of energy within us. I am very much thankful to co-convener sir, Professor Dr. Ganesh Prasad Mishra ji and Mr. Prasenjit Paul ji for their guidance. I am very much thankful all the departmental and interdepartmental faculty members to be present here. An event of this dimension not happen tonight, the wheel start rolling month in advance. We have been fortunate enough to be backed by the organizing committee of very motivated faculty members, non-teaching staff members and volunteers. Thank you for with us. I am very much thankful to our association, Internal Quality Assurance Cell, IQAC, and Indian Pharmacy Graduate Association, IPGA. It would have been impossible to organize it without their support. At the end, I would like to thank to Marcom, IT, PPT, media, and other auxiliary supporting departments. If time is a money, then today you all spend millions of for us. Thank you all of you for making this national conference successful. सभी को आयोजक टीम की तरफ से ढेर सारी शुभकामनाएं. Thank you. जय हिंद.
Thank you, Dr. Manish Pathak, sir. I requesting to all volunteers of the discipline, please come on the stage. And I also requesting to Dharmendra sir and Deepak sir, please you are also come on the stage for one photography. Thank you. आईफोन वाले ही फोटो खींचेंगे बस